Biology Part 177. This produces secondary phloem and xylem in the continuous bands. As secondary xylem is laid down in the layers to the inside of the diameter of the stem increases, and the cambium, cylinder, and secondary phloems are pushed outwards, the inner core of the xylem is known as wood. In older stem annual rings can be observed. These appear because the new xylem vessels formed in the spring are large and thin-walled, while those formed in the latter in the autumn are smaller and possess thick walls. Most but not all the xylem cells are arranged in the vertical columns. Here and there, horizontal parenchyma cells form the structures called rays. These are arranged radially and are living channels that allow the metabolic byproducts and substances like the resin to be deposited in the wood. The epidermis forms the outermost covering in a young shoot, but as stems increase in diameter, old tissues split and are lost from the surface. Well, the production of older stem is achieved by the production of coke. Coke tissues consist of a small cell with cell walls impregnated with a fatty substance called the suberin, which waterproofs these cells and makes them impermeable to gases. The coke is formed by coke cambium, which is derived initially from the outer layer of the cortical cells. As the stem continues to increase in the girth, the older layers of the coke are ruptured deeper tissues form a new cambium. In many trees, coke is formed by the coke cambium derived from the parenchyma of the cells of secondary phloem. The bark of the tree consists of all the tissues outside the vascular cambium, and that is coke, the coke cambium cortical cells if present in the secondary phloem. The gas exchange while living tissues of the older stems will be complex except for the presence of structure called lenticels, which penetrate the outer coverings. These consist of loose parenchyma. Cells allow the inward and outward diffusion of the gases at the expense of some inevitable water loss. And now we talk about the adaptations for roots and leaves. Well, roots development, the tutorial will describe the structure of the roots and leaves and outline their important functions with the exception of the uptake and transport, which are described in the later tutorial. The structures present near the tip of the growing root are illustrated, and they show the appearance of the radicle of the germinating broad bean. Simple experiment in which the radicle is marked with an ink revealed that the elongation occurs in the region just behind the tip. The region nearest to the tip consists of the meristematic cells, while older cells beyond the region of the elongation shows up the signs of differentiation, as is the case of epidermal cells with developed roots hairs. Unlike the tip of a shoot, which is protected by small leaves inside a bud, the tip of a root is protected by a special structure called a root cap. The apical meristem within the root tip gives rise to three primary meristematic tissues corresponding to the shoot. These are the protoderm, ground meristem, and the procambium. In addition, the apical meristem gives rise to the cells of the root cap, which are continually renewed to replace the cell lost at the surface as the root pushes between the soil particles. At the center of the apical meristem is a region of cells known as the quiescent center. These cells divide extremely slowly and appear regulate root growth while limiting the number of rapidly dividing cells or initials which form the remainder of the meristome.